Hello everyone, I'm Amir Syazwan, and together with my friend Najman Naqib, we will be presenting on the structure of the mature HIV-1 CA hexamer in intact virus particles, commonly referred as the 5-MCH protein. The 5-MCH protein structure represents the mature HIV-1 capsid hexamer, a critical component of the human immunodeficiency virus or HIV-1. It plays an essential role in protecting the viral RNA genome and mediating interactions with host cells during infection. The structure is stored in the Protein Data Bank (PDB) under the ID 5-MCH. It was discovered by a research team that was published in 2016, provided high-resolution insights into the architecture of the HIV-1 capsid, advancing our understanding of its role in viral replication and potential therapeutic targeting. The 5-MCH structure consists of 26 protein chains, all forming part of the capsid protein or CA. These chains are arranged to create the hexameric and pentameric units essential for the conical shape of the HIV-1 capsid. The CA protein is composed of two main domains. First, the N-terminal domain, NTD, and the C-terminal domain, CTD. The NTD forms the core of the hexamers, contributing to the capsid stability. Meanwhile, the CTD stabilizes the lattice and connects adjacent hexamers, ensuring structural integrity and adaptability. When we look at the secondary structure, the model shows an arrangement of alpha helix and beta sheets, which are characteristics of the capsid protein. It's important to note that 5-MCH lacks active sites, as its primary role is structural, not catalytic. 5-MCH structure was determined using cryo-electron tomography or cryo-ET combined with subtomogram averaging. This advanced imaging technique allowed the structure to be resolved at a resolution of 6.8 Armstrong. This makes it one of the highest resolution models of the HIV-1 capsid in its native environment, providing unprecedented insights. Understanding this structure is essential because it reveals how the capsid assembles and functions, a crucial step in the HIV life cycle. These insights form the basis for developing antiviral targeting capsid assembly or disassembly. 5-MCH's main function is structural, encapsulating and protecting the viral RNA and enzymes during infection. It is not catalytic and does not have active sites, as its role is to form a stable and adaptable lattice. Unlike enzymes, the capsid lacks regions designed for catalytic reactions. Its role is to purely structural, supporting the virus life cycle by maintaining genome integrity and aiding in host cell interactions.
Alright, so the alpha helix is a right-handed coil formed by amino acid residues in the polypeptide chain, which is responsible for the structural integrity and stability of the protein. Hydrogen bonding between the carbonyl oxygen of one amino acid and the amide hydrogen of an amino acid four residues earlier in the sequence give rise to the alpha helix. Even though no hydrogen atom were mapped in this particular protein due to their low electron density, it does not mean that the hydrogen does not exist, because amide is an essential part of amino acid, which is the makeup of all proteins. Moving on, the formation of beta sheets in the protein originated from the hydrogen bonding that occurs between the backbone amide NH of one strand and the carbonyl oxygen C-double-bond-O of an adjacent strand, stabilizing the sheet structure. Beta sheets can participate in important intermolecular forces in an interaction that are fundamental for protein-protein interaction, quaternary structure formation, and aggregation process. This is particularly relevant for understanding how HIV interacts with host cell factors during viral assembly. This protein consists of 26 polymeric chains, with each chain composed of a total 833 atoms and 221 residues. The atoms making up the structure are identified as carbon, nitrogen, and oxygen of equal value for all chains, and no hydrogen is mapped. However, as I mentioned previously, this does not mean that hydrogen is not present at all in this protein, but it is considerably more convenient to exclude them in the mapping specifically due to their low electron density, which will be this protein consists of 26 polymeric chains, with each chain composed of a total 833 atoms and 221 residues. The atoms making up the structure are identified as carbon, nitrogen, and oxygen of equal value for all chains, and no hydrogen is mapped. However, as I mentioned previously, this does not mean that hydrogen is not present at all in this protein, but it is considerably more convenient to exclude them in the mapping, specifically due to their low electron density, which will be. Next, to know about the quality of the chain previously discussed, we can take a look at the residue property plot. The first graphic, the straight line, summarize the proportion of the various outlier classes displayed in the second graphic. The second graphic, meanwhile, shows the sequence view of the outlier. Residues are color-coded, in which green mean no outlier, yellow one, and so on. So, for example, for chain A, there is 9% red residues with 98% green residues, showing that it is a high-quality chain.
The N-terminal domain, NTD, of the MCH structure plays a crucial role in the formation of hexamers. It forms the central ring in the hexameric lattice, which is essential for maintaining the structural integrity of the HIV-1 capsule. Structurally, the NTD is composed of alpha helices, which provide rigidity and stability to the capsule. Additionally, it is linked to the C-terminal domain, CTD, by a flexible connector, allowing the lattice to curve and adapt to conical shape of the capsule. NTD adjusts its geometry when incorporated into pentamers, enabling the capsule to curve and form its conical structure. This flexibility is essential for creating the closed, stable capsule that protects the viral genome. The NTD also has potential binding sites for host factors, which play a role in viral transport and infection. These sites allow the capsid to interact with the host cell machinery effectively. The N-terminal domain, CTD 5MCH, plays a crucial role in lattice stability by connecting monomers and stabilizing both hexamers and pentamers in the capsid structure. It is linked to the N-terminal domain, NTD, through a flexible connector, allowing the capsid to curve and adapt to its characteristic conical shape. This structural flexibility provided by CTD is essential for the adaptability of the capsule, enabling it to achieve the precise curvature needed for its function. The CTD also maintains connectivity between hexamers and pentamers, supporting the overall assembly of the capsule and ensuring its structural integrity.
The study of the HIV-1 capsid hexamer has significant application for drug design, mutation mapping, public education, and bioinformatics advancement. By understanding the structure and function of this viral component, researchers can develop effective antiviral therapies, personalized treatment plans based on mutation patterns, enhance public awareness of HIV, and foster collaboration in combating the virus. These efforts contribute to better health outcomes for individuals living with HIV and AIDS and address ongoing challenges associated with the virus.
In conclusion, the 5-MCH protein representing the mature HIV-1 capsid hexamer plays a critical structural role in the life cycle of HIV-1. Its key functions include protecting the viral RNA genome, facilitating interactions with host factors, and ensuring proper transport and infection. Structurally, the NTD and CTD work together to form and stabilize the hexameric and pentameric lattice, providing the necessary flexibility for the conical shape of the capsule. The discovery of 5-MCH using advanced techniques like cryo-electron tomography has significantly deepened our understanding of the capsule assembly and function. These insights pave the way for developing targeted antiviral therapies and new strategies to combat HIV-1 infection. That's all from us. Thank you for your attention and have a nice day.